Hi all, Dr. Clark here again. For Gen Bio today, we're going to continue talking about genetics, but now we're going to talk about chromosomes, Mendelian inheritance, and things that might affect that, like age of the organism, and incomplete separation during metaphase of the sister chromatids, and things like that. So first I'm going to show you a video of an event where, this green right here is a pair of chromosomes, homologous chromosomes, that are they've been had a fluorescent dye infused onto them, and you're going to see that they do not separate during mitosis. In this case, this is meiosis, but they're not. So these are sex chromosomes, but these are sex cells. Sorry, not sex chromosomes. They're not going to separate, which is what we call nondisjunction. And so I'm going to show you a little video so you can see how that works. So hopefully that was pretty clear, that you could see. We're going to watch it again real quick. That single cell had both homologous chromosomes. So that results in a fewer number of chromosomes in this cell and a larger number of chromosomes in that cell. Okay? That's called nondisjunction. And so we're going to look at what that means for humans, for maybe diseases, disorders. We're going to talk about some situations like that. And we're going to talk about that connection between Mendelian inheritance, so Mendel's law of segregation and independent assortment, and its link to chromosomes. All right, so first off, remember that Mendel was working in the 1850s, in the mid 1800s, 1860, and through into the 1870s and whatnot, is when he was doing most of his work on genetics. And there was a long lag period. I already talked about this because he published things in obscure journals. But in the 1900s, early 1900s, Walter Sutton came around. And I think in the previous video I called him William Sutton, okay, but it's Walter Sutton. Sorry for that. But he came around and did some research to figure out what are individuals actually inheriting. Okay, so Mendel was saying they're inheriting characteristics. We later said that these characteristics are genes, but what's the actual mechanism of inheritance? Okay? And so Sutton was working this out, okay, and he started looking at reproductive cells and found out that when reproductive cells divide and you actually look at sperm and egg, especially in diploid organisms, they only have one copy of the genetic information. So they only had one chromosome. For chromosome number 1, 2, 3, etc., they had one copy of it. And he later, extrapolated this. Well, that is due to meiosis, or what we call reduction division. So the original number can return. So the gametes can then fuse and return the original number. And so his research came up with that Mendel, when he was talking about segregation of genes, how genes are not associated with each other, so the inheritance of one gene is not dictating the inheritance of another gene, and independent assortment, that you only get a certain number of genes, okay, and you're only getting one copy in the sperm and one copy in the egg, that it wasn't the genes that were doing the segregation. It wasn't the genes that are doing the independent assortment, but rather it was chromosomes during meiosis. Okay? And so it was really Walter Sutton's work that showed that chromosomes are the carriers of genes, the vehicle of the gene. Later, Thomas Morgan, who we talked about before because we talked about Thomas Morgan in regards to crossing over. Remember, Thomas Morgan is one of the first people to write about crossing over. Okay? His work was on inheritance of chromosomes again, but now he discovered something else that's also very important for sexually reproducing organisms. So Morgan was studying Drosophila melanogaster, which is the common fruit fly, and still today in labs, okay, 100 years later, we still are using Drosophila melanogaster for a lot of genetic research. They're a great, laboratory organism. They're easy to study, they're easy to grow, they're easy to maintain, and we know a lot about their chromosomes and their DNA for that matter. They've been genomed, so we know all their genes, and we can clone them. We can do all kinds of things with them. Okay, but at the time, not much of that was known. But Thomas Morgan was still using Drosophila melanogaster to look at chromosomal inheritance. So what he did in 1910, he noticed that there was a mutation in male fruit flies. Okay? So a normal fruit fly has red eyes. And you've probably seen fruit flies before if you've kept fruit in your house. Occasionally fruit flies will magically appear. If you ever look at one really close, they will have red eyes. Okay? But there is a mutation that can occur that gives male fruit flies white eyes. So what Morgan did is he tried to figure out, well, does the inheritance of the white eye follow a Mendelian pattern? Can you predict it based on a Punnett square? So in order to do that, he had to do some crosses, just like Mendel would have. But now he's working with animals. And so he crossed a mutant male, white-eyed male, with a normal female, a red-eyed female. And as you would predict, the eye color, okay, in the F1 generation was all red. Okay? Every single fruit fly had red eyes. But in the F2 generation, males had white eyes and no females had white eyes. So only males had white eyes, and there was never a white-eyed female. And so this was of interest to Morgan. So he started to look at what's going on at the chromosome level. And what he figured out was sex was the key to the results of this cross. That in flies, just like humans, two X chromosomes, two of their sex chromosomes, are X chromosomes, is a female, and a male is an XY. Right? Just like humans. Okay? Now, Morgan said that the white-eyed trait must reside on the X chromosome, right? The sex chromosome. And that is why you only see it in a sex. And so you might be saying to yourself, well, then why can't females get it? Why don't you see it in females? And the reason for this is that typically the double X, if both X's have a mutation, the female wouldn't even produce eyes, and the female would actually die. Okay, so females never had it, but there's enough variation that if you had an X with a mutation and the Y doesn't carry that mutation, it doesn't carry that gene, that the male would produce this weird variety of having white eyes, okay, and not the typical red eye. So we call those sex-linked traits, or sex-linked chromosomes, sex-linked genes. Okay? And we know lots of these, or we assume lots of these, things like color blindness, which is a sex-linked trait, and other things. And we'll look at these as we progress through the remaining lectures. Okay, so his experiments demonstrated that there was such thing as called sex linkage, which has to do with the sex chromosomes of the organism. And if you take that into account, the sex of the individual, then indeed, right, the white eye variety versus the wild type, the red eye variety, is a Mendelian trait. It fits the Mendelian Punnett square, Mendelian inheritance. It also explains the law of segregation, that traits are independent because chromosomes are independent of one another. Okay? And so this was just more evidence suggesting that chromosomes are the vehicles for the genes to sit on. Okay? Now we now know that there's even more to this. Okay? So you can look at a single chromosome. So these are homologous chromosomes. Let's say this one here is chromosome from dad, so this is the paternal line, and the bottom one here is chromosome from mom, the maternal line, and we're talking about pea plants. Now, pea plants will have many different genes that are on this chromosome that do different things. Some of them are going to be related to phenotypic variation that you can physically see. This is what Mendel was working on. So things like flower position, pod shape, plant height, etc. Okay? There's going to be a lot of other genes that Mendel couldn't see. And these would be genes that are coding for many different things, like proteins in the electron transport chain, thylakoid membrane proteins, mitochondrial proteins, lots of different things. Okay? All kinds of different things could be also laid in between these two things, or these things. But Mendel was picking up on these physical characteristics that you can actually physically see. Okay? So in some situations, you have things like flower position, which is off by itself on the chromosome and never really the result of or limited to crossing over. Right? These are not linked to anything. So maybe it is linked to it could be linked to a protein that's embedded in the thylakoid membrane, but it's not linked to another trait that you could physically see. But some things develop what we call linkage, which means they occur next to each other on the chromosome. So like pod shape and plant height. So maybe this is, a plump pod and a tall plant, okay, versus a constricted pod and a small plant. Well, these two variables, because they're linked together, it doesn't matter how crossing over occurs. Right? So remember crossing over from meiosis during prophase, you get this crossing over event. These are, because they're linked, they never break. So you always get plump pods and tall plants. Always. Because they're tightly associated with each other. Alright, so let's look at some human chromosome stuff, okay, and how humans are subjected to the same laws that all other organisms are subjected to when it comes to Mendelian genetics or really anything on the planet. Okay? We have 46 chromosomes, okay, which means that we have 23 different chromosomes and pairs of each one of them. 22 of the 23 pairs are called autosomes. And there's really no real difference between the male and the female. At least there's no difference in centromere placement and no difference in the size of the chromosome. Now there's a difference in the actual genetic code in some of the genes, which we call allelic diversity. So the alleles are going to be a little bit different. Okay, but the genes that code for different things on each one of those chromosomes are identical to each other. The last pair of chromosomes is what we call sex chromosomes. And sex chromosomes for humans, okay, are going to be in the form of XX for a female or XY for a male. Now remember, if we're talking about different species, there's a very different variety, and we already talked about this before, but like if you're talking about birds, it's ZW and ZZ. If you're talking about fish, it could be ZWZZ, it could be XXXY, it could be XXXX, and there's a lot of variety. So remember, sex chromosomes differ based on the organism, and we don't have a good grasp on all the varieties. We know that there's a massive amount of varieties throughout the animal kingdom. Now, the interesting thing about human chromosomes is that Y chromosome gives males their unique traits. Okay? And so that's what make males male. So the maleness, I guess, for that individual. So things like Adam's apple formation, high levels of testosterone, lots of different things that make a male and not female. Production of testes and things like that all occur on the Y chromosome. Now, during meiosis, we talked about this before, sometimes there's mistakes that happen, right? Mainly during the phase at which either homologous chromosomes are separating or sister chromatids are separating. Okay? So metaphase, either metaphase 1 or metaphase 2, is really kind of the result of nondisjunction. So what's going on is when those chromosomes line up on the metaphase plate, equatorial plane, sometimes the microtubules don't attach to them correctly. Or sometimes the microtubules might attach, but the forces are not enough to separate the cohesion that holds them together, or to break the centromere that holds it together. So they separate incorrectly during metaphase 1 or metaphase 2, moving into anaphase 1 or anaphase 2. That's called nondisjunction. When this occurs, it forms what we call aneuploidy, okay, which means you have a abnormal or a different number of chromosomes in some of your cells. Now, when this occurs in somatic cells, which it does, it occurs in somatic cells, a lot of times those somatic cells will either just be moved straight into the GO phase or go through apoptosis. Now, when it occurs in sex cells, in germline cells, a lot of times, if that germline cell is fertilized, it'll result in death, right? Because you just have too many chromosomes. But there are certain situations, there are certain places where that can occur. And so we're talking about nondisjunction in meiosis, sex cells, okay? And what happens with these situations. In mitosis, nondisjunction can still occur. Okay, you can still have a different number of chromosomes in one cell versus the other. It's just normally those cells are dealt with by pushing them into a GO phase or destroying them. But in meiosis, a lot of times they're allowed to persist, and if they actually are the ones that fertilize or are fertilized, either death occurs or you have some kind of abnormality that it occurs in the individual. Now, some of these we know a lot about, okay? Like chromosome number 21. So in chromosome number 21, if instead of the normal 2 copies, instead you get 3 copies, okay, this results in Down syndrome. Okay? And so that's an extra copy of chromosome number 21. Now, this nondisjunction occurs in the case of females. What's occurring is that females, as they age, and we're going to look at this in a second, but as they age, their chromosome number 21 becomes more sticky. Okay? The synapses between the homologous pairs become more tight, and they don't release, they don't break apart as a female gets older and older. And so as you age, the likelihood that nondisjunction occurs for chromosome number 21 increases, okay? And it increases drastically. Okay? You can also have this occur in chromosome number 22, which can cause triploidy or cat eye syndrome, right? Which can have lots of different adverse effects, like the pupil of the individual is kind of more linear, or runs kind of in a vertical pattern, kind of like a cat, but it's not like a, it doesn't look just like the cat pupil. It's kind of more blurred. That's one thing which causes the vision of the individual to be poor. But there's lots of other things that can occur. You can have poor kidney function, poor liver function, etc. if you have 3 chromosome number 22s. Okay? Now, the individual can survive into adulthood, though, just like Down syndrome. And so here's what's going on. So we talked about this before. Okay, so you have metaphase 1, these homologous chromosomes are lined up, and then right afterwards, these homologs should be separating. Here you can see the microtubule did not connect to that kinetochore, or to that centromere. So that kinetochore never connected to that centromere. So the homologs never get separated, and they get pulled over here. Then they get separated after metaphase 2, but you already have too many copies of this chromosome in each one of these cells and not enough in this cell, which results in aneuploidy, okay? And can result in the fertilization of one of these two. So if these are sperm, they could make it to an egg. If these are eggs, they could be the one that it becomes mature and is fertilized, and therefore the individual, after fertilization event, they would have 2 copies of whatever this big chromosome is. We could call it chromosome number 1. So they would have 3 copies of it. They have 2 in this cell, and then the sperm would add another copy. Okay? And that's what's going on with Down syndrome. So here is a karyotype of someone with Down syndrome. You can see chromosome number 21 there, and you can see they have 3 copies of chromosome number 21. Now, depending on what genes are turned on and a few other factors with other chromosomes that are going on, will depend on the severity of Down syndrome and the functionality of that individual. But nonetheless, if you have 3 chromosome number 21s, you will have Down syndrome. You develop Down syndrome. Okay? Now, here is a graph showing the age of the mother, okay, and the likelihood of producing a child with Down syndrome. Okay? So young females, the likelihood is poor. Okay? We're talking all the way up to age 30, even close to mid-30s. The likelihood of producing a child that has Down syndrome is about 1 out of every 1,000 births. Okay? As you increase age, okay, it drastically increases. The stickiness of those homologs increases, and the likelihood that they do not get separated increases. All the way to when you're out at 50, you have basically a 1 in 10 chance, a 10% chance that you'll produce a child with Down syndrome. And that actually increases even further the further you go past 50. Okay. Now, nondisjunction can affect sex chromosomes also. So that's autosomal chromosome 21, 22. There's a few other that can form and have offspring be developed from those. Most of the time, though, if you get like 3 copies of chromosome number 1, that embryo never develops. That zygote never develops. Okay? There are a few situations where the zygote will develop into an offspring, and the individual can live into adulthood. Sometimes they only live for a few years, okay? But sometimes they live all the way to adulthood, and they can reproduce in some cases on their own, and reproduce that, have that chance of passing on that characteristic to their offspring. But nondisjunction can occur in the X chromosome, okay? And we can get variations. So you can have triple X females, and it depends on again, what genes are turned on and whatnot, but there's a lot of different symptoms that can occur. Okay? Most XXX females are going to be average or above average in height and body mass. So, you know, I don't know for a fact. There's some hypotheses that females that are extremely tall and don't have a connection to tall individuals in their family might be XXX females. But really, you can live a perfectly good life and the person next to you could be a XXX female and you would never know, and it really doesn't affect them very much at all. Okay, they're not sterile, they can reproduce offspring perfectly fine. Now, you can get a male that gets 2 X's from a female and then the Y from the male, from the sperm. Okay, and this is going to be Klinefelter syndrome. Okay? This is a sterile male that will have female features. Okay, the Adam's apple will be not visible. They might develop breasts. It really depends on the the severity and what genes are turned on and what genes are turned off. But they'll have many female features, but they'll still be male. You have Turner syndrome, which means that they get an X from the male, but nothing from the female. And this is going to be a sterile female. Life expectancy of someone with Turner syndrome is about 20 years. They're gonna be very diminished stature, very short. They're gonna have what we call a web neck, which means they'll have kind of skin, loose skin in the neck region, which indicates that certain parts of their body grow fast but don't fill in, so they can have loose skin in other regions and things like that. But the individual will be sterile. And so this is non-disjunction when it comes to females. This is what happens when the female gamete, the egg goes through meiosis and doesn't separate. The sex chromosome doesn't separate, so she gives 2 X's in one egg, nothing in the other. The male is normal, so they can make a XXX female, Klinefelter male, Turner syndrome female, or nothing, non-viable if you just get a Y. Okay? It also occurs though when non-disjunction happens with the Y chromosome. So this is not just a female thing, but males, it can happen to males also, where the male is giving 2 Y gametes. Okay? And so some cases you get this XYY, which these males are fertile and have normal appearance, but there is a little bit of evidence that suggests that these males are over-aggressive. Okay? There has been some chromosome examinations in other countries that suggest that violent males might have an extra Y. Now, I don't know how much of this is just correlation and not really anything that has to do with having extra Y's, but nonetheless, that data's out there and you can look it up. Okay, with that, that's really the role that chromosomes play in dictating genetics. Okay, so we now know that genes reside on chromosomes due to Morgan's work and Sutton's work, and that you can have variations of genetic disorders based on aneuploidy or non-disjunction, where chromosomes don't separate correctly. And we'll look at some other situations where chromosomes might, and not just the separation works, but the crossing over might change, and the crossing over might be faulty. Okay, and we'll talk about those a little bit next time. But next time we'll also hit on pedigrees. Okay, so how we can look at a line of individuals and try to figure out what or who passed on a certain genetic characteristic, okay, without looking at the individual's DNA. All right, so next time we'll concentrate on that.